Okay, I'm going to talk about the viruses. It's a really pretty short section. We're basically looking at 19.1 and 19.2, and then I mentioned this in class, but also relating it back to very beginning of the semester with the themes from chapter 1. So viruses, we've talked about cells in this unit and talked about their single, viruses are single units, like a cell is a single unit. And they have certain parts, but are missing other parts. So we're going to see kind of like how is a cell built and how is a virus built, and both structurally and the actual literal structures that are being built and how they're being built are both going to be different. And so we can see about considering viruses alive or not. So here is a picture, these little viruses. This is a bacterium, so it's E. coli, and these yellow guys, little, sort of look like some kind of screw or something like that. Those are bacteriophages. They're viruses, and they're invading or infecting this bacterium, and then they will replicate, and the bacteria will break open, and then hundreds of them will be released to go on and infect more cells. So bacteria get viral infections just like all other organisms. Okay, so here's our objectives. I pretty much touched on these, and you should go back and look at this sheet to make sure you understand these concepts. But first we're going to talk about the components and how they're the same and different as living organisms. So we don't actually consider viruses alive at this point. It is somewhat debated because they do have a lot of characteristics, but the real main problem is that they can't replicate on their own. They have to have a host. And it's not just have a host for like, oh, I need somewhere to live and nutrients, and you get your nutrients by being a tapeworm in the intestine of another organism. It's literally like they don't have the components in their cells to make a copy of themselves. So we think of them more as a collection of chemicals that have somewhat living-like functionality, but they're not considered living because we call them obligate parasites. Anyway, the obligate parasite means that they need a host. So they're not cells. They're very small, infectious particles. So there's two main parts all viruses have. The inside of all these pictures, either red or blue, that is their nucleic acid. So it can be DNA, it can be RNA, it can be double-stranded DNA or RNA, or single-stranded DNA or RNA. So a lot more variety in the genetic material of a virus than there is of living organisms. The nucleic acid, whether it's DNA or RNA, is then enclosed in what protein coat. It's this purple. Remember, our proteins are purple in this book. So these purple parts, and it's basically made up of repeating subunits. And then sometimes, in this example here, you can see the yellow part, that's a lipid bilayer envelope, which actually is surrounding the outside there. And you can see sometimes they have real fancy construction of their external, the protein coat, the capsid there, and sometimes it's much more simple like a rod. So here's the parts, like I just said, the capsid made of little units called capsomeres, capsoparts, right? Mere is part. So there's all these capsomeres, and they can be built into all different types of structures. They're protein. There are modifying glycoproteins on the outside of some of these viruses, and you can see what they'd look like actually in micrographs here. So we have tobacco mosaic virus infects the tobacco plant. I think almost all, if not all, tobacco plants today are infected with it. I mean, it doesn't hurt us at all, but it is bad for the crops. Adenovirus, we have the influenza virus, and here's bacteriophage. So here's the tobacco mosaic again. This is the rod-like kind of structure there. That's the adenovirus. It's icosahedral. That's a bunch of different faces. Very geometric. Here would be the envelope I was talking about. So the viral envelope in, for example, in the influenza virus. It's useful for the virus because the envelope can display proteins on the outside, glycoproteins. Remember those embedded membrane proteins with carbohydrates on them? And they can have some of them that are from the host membrane, and so it's easier for the virus to infect another cell because it can somewhat trick, if not completely trick, the host cell into thinking that it is self and not an invader because it's cloaked itself basically in the right proteins. Bacteriophages are structurally pretty complex. You can see they have a head and a tail and all kinds of parts that are going to do different functions. And we can also call them just phages, or I hear sometimes phage. And these guys have been used to research a lot about the DNA in the early days, lots about all kinds of molecular biology, because they're simple enough and that they're made of just protein and just DNA. And we'll see these again in chapter 16. So here they are. You can see there's the tail. Okay. Viruses reproduce. Like I said, they're obligate intracellular parasites, meaning they must have a host. And like I also mentioned, they need the host for actual parts to actually do the replication. So they can't make their own proteins, and they can't, a lot of viruses can't make their own nucleic acid either. So basically they come in and they take over all the things that are needed to make, transcribe, translate, replicate, DNA, proteins, all that good stuff. So they use all the enzymes from the host, the ribosomes, tRNAs, amino acids to build those proteins, to build the capsids, and ATP. And then once the parts are made individually, they actually self-assemble into new viruses, which then can leave the cell and go infect more cells. So you see this here. This is a virus with DNA in the inside, so the coat goes away, or the coat breaks off from the viral DNA in this case, so the DNA protein are going to separate. Some of that DNA is replicated in order to make new genomes. Some of it is read and made into mRNA. It's transcribed, and then that's read and made into protein that's translated, and this would be done by the ribosomes, the step here making the protein. Those capsids are then put back, or spontaneously assemble around the DNA, and then the viruses are ready to go. I'm not going to show you the videos. They are on your Mastering Biology. I highly suggest you watch them on there. As far as phages go, that's these guys, they have two reproductive cycles. There's one we call the lytic cycle, and there's one we call the lysogenic. So lytic, I'll go back to lysogenic in a minute. Lytic means to break, like hydrolysis or lysosome. All these are lysing. The cell lyses when it's in a hypotonic environment, an animal cell. So these lytic cycles is basically going to end up with the host cell bursting open and releasing all the new phages. And these phages, bacteriophages, infect bacteria, which you'll see later. Actually, we get molecules that we use for biological research that have been instrumental in understanding genetics and basically almost all the kind of modern biology we understand. We actually get them from bacteria, and bacteria have them to protect themselves from viruses because they actually cut up viral DNA in order to keep the infection from taking over the cell. we've co-opted them for our own, research and medicine purposes. So here's the deal. So the attachment happens. The DNA goes into it's injected into the bacteria cell. The host DNA is chopped up. The synthesis of viral genomes and proteins, it self-assembles, and then the lytic cycle it breaks open or digests really, the cell membrane and the cell wall, and then it will be released from the bacteria. And then each of those guys can go on to infect another bacterium in the population. We're talking hundreds of these are released at once, not just, 5. So that's called the lytic cycle bursting open. The lysogenic cycle, genic, you can think genes, because this is going to be the cycle where the viral genomic material, the viral DNA, hangs out in the host genome for an extended period of time. Doesn't kill a host cell, at least not right away. If it's in the lysogenic cycle, it's not killing the host cell. So what happens is you have, start here. So what would happen would be the DNA would be injected from the phage into the bacterium. So here is your viral DNA. If it goes in the lysogenic cycle, which is what we're looking at, it's going to get incorporated. The viral DNA gets incorporated into the bacterial chromosome. You can see it's circular chromosome. So here now it's called a prophage now. And it basically just gets replicated along with the bacterial DNA, just like normal. And so every new bacterium you make via binary fission has that viral DNA sequence in the genome and just keeps going. Now, this is, cells aren't bursting open, so sometimes you don't get as sick from these, but on the other hand, there could be genes being read off the viral DNA that are producing certain chemicals and toxins that could be making you sick. So just because your cells aren't bursting open, and obviously that would make you ill, they could just be completely dormant, or sometimes there's actually expressed genes that could make you ill anyway. And a lot of these also, if they are sometimes they are dormant and they're reactivated under certain environmental conditions. So people who get viral infections and then you say, oh, they never really go away, like chicken pox, varicella zoster, never really goes away, can come back later as shingles. Same virus, different disease because of your different age, and some of the symptoms are different. But really, the virus just hangs out, incorporated or sometimes next to it, but hangs out in the cells, keeps on getting replicated, and then eventually causes problems again, possibly. Some phages can go between both, so you see here's the lysogenic cycle we talked about, and certain environmental factors can bump it into the lytic cycle, where it comes out and it does package itself into new phages, and those explode or burst open the host cell and then go infect more cells. And then it can either keep on going the lytic or go back into lysogenic. And the cells that do this kind of, viruses that do this kind of cycle are called temperate phages. And it's really environmental signal dependent. And then finally, I mentioned this, is that we want to look at the different characteristics of life and see how viruses fit and don't fit these categories. So, for example, something like order, you're thinking, are viruses totally random? Or is there some type of structure, a consistent structure to their physical makeup? If there's structure to it that has repeating subunits in a specific pattern, particular molecules in particular places, that's all order. If it's a jumble of random accumulation of molecules, then it's not going to be ordered. Reproduction, we've discussed, that's a really important one. And response to environment, I just mentioned. Evolutionary adaptation. If you just take a look at this chart, I'll just show you. This is talking about treatment with this 3TC drug, and it treats the HIV, so that's a virus. And it works really well at the beginning, but then the viral population became resistant very quickly. And so what they found out happened was, if they tracked it, that within, depending on the patient, but within, 4 weeks, or I guess 8 weeks, within 8 weeks, within 2 months, 100% of the viruses within patient 1, 2, and 3 were all resistant to 3TC. They weren't being killed by it anymore. And it's not because those viruses, felt the drug and decided they needed to figure out some way not to get killed by it, right? Because you can't change the genes you have. What you can change, or what does happen, is the population can change. So there was a virus in the population, and there's a lot of variety, lots of genetic variation, and it had some kind of characteristic, some genetic combination that allowed it not to be killed by this 3TC. And in fact, we can actually go and look. I don't have it for this one, but we can go and look, and I'll talk about this at the end of the semester again, and see, well, what gene mutation do they have, and how did that allow them to get around or allow them not to be killed by the 3TC. And you can see this is a pretty fast. I mean, patient 1's resistant within 2 weeks. And so, clearly this is a population that's evolving, which means changing genetic makeup over time. And that's why we have such a hard time fighting viruses off, bacteria too, for that matter, because they have a very fast life cycle. And viruses also have a really high mutation rate because they don't use proofreading like living cell organisms do. So that's a nice evolutionary adaptation example. And so you should think about growth and development, meaning going from, an early form of that organism, like a single fertilized egg or something, into a functional, multicellular or coordinated kind of organism there. Energy processing, so taking in external energy in the form of like carbon usually. Regulation, so here there's things like thermoregulation, as well as like water retention regulation, so kind of like something with the environment, where the environment influences how much or how little of exchange goes on with the environment. So just think if viruses do these things, and have some of those examples ready to write about of what they do have and what they don't have. Okay? That's the part of chapter 19 we're going to want to know. Okay, so good luck.